So thank you, Mazen. Thank you, Dr. Ali. Thank everybody at Kasaralaini. It's a long friendship now, and it's always an honor for me to come back to Cairo. Those who know me know how much I like it coming here, and I'm always grateful for any invitation. The topic I've got today, or I was given today, is a little bit unusual for me because, as you know, I'm an endoscopist, and I'm talking about a topic I had to learn a lot about, because this is a lot about basic science, but it's, I think, one of the most fascinating topics in gastrology today. So what are we talking about when we talk about organoids? As you know, the GI tract is a continuous structure. It starts at the mouth and ends at the anus, and we can divide the digestive part of the GI tract into the stomach, the small intestine, and the colon. I mean, we all know that. We also know that the organs are in constant contact with nutrients, with metabolites, with bacteria. So that's a very challenging environment, and these interactions are indispensable for our health. But on the other hand, there is constant damage. We are confronted with a lot of toxic substances. There is DNA damage which has to be repaired. So there is a continuous self-renewal of the epithelium throughout the GI tract to minimize the damage which would otherwise accumulate and cause cancer, for example. And this is achieved mostly by adult stem cells, which are residing in spatially restricted parts of the GI tract, of the tissues, small bowel, for example. And they continuously divide and give rise to progenitor cells, which then mature into the tissue cells of this particular organ. And you can say that every 3 to 5 days, there is an entire renewal of the cells of our GI tract due to these mechanisms. And this is orchestrated by a very complex network of signaling pathways which control this renewal and make sure there is no malignant developments. But of course, in case of deregulation of this balance between activation of growth and suppression of growth, there is the risk of malignant transformation, there is the risk of cancer. So it is crucial for our understanding of the GI tract to understand those processes regulating stem cell homeostasis. And the impact, that this is not just theoretical, the impact is very important. You just have to look at cancer statistics. And if you look at worldwide cancer statistics, the 3rd and 4th most frequent reason for cancers in the world are, and cancer death most importantly, are stomach cancer and colorectal cancer, just to give you the importance of this issue. So, organoids, what are they? They are 3-dimensional compared to cell cultures, and they are miniature structures cultured in vitro, produced from either human pluripotent stem cells, PSCs, or adult stem cells, ADSCs. And they are derived from either healthy individuals or patients, and they recapitulate the cellular heterogeneity, structure, and function of human organs. So they are sort of miniature organs, at least in part. We used animal models and cell cultures for a long time in medicine to improve the understanding about the way the human body works, to improve our knowledge of cellular signaling pathways, for developing guidelines for the design of drugs, for identifying potential drug targets, and for clarifying mechanisms leading to various diseases. The value of those models is undiscussed. We've used them for many, many years, and we're still using them today. But, as you can see in this cute little mouse, there are clear differences between humans and animals, so not all the findings in animal models can be translated into the human body. And if we're talking about cell cultures, we have two-dimensional cell lines. There is a lack of hierarchical structure, a lack of 3 dimension, lack of cellular diversity, They're all monoclonal. And the cell and cell matrix interactions are lacking. So that's why we can't really have a clear comparison between the functions of organs and those cell cultures. So the advent of pluripotent stem cells technology and adult stem cell technology changed a lot because it opens the pathway or the streets for in vitro human-specific models of various diseases, of genetic variations, of cancer, for example. And recently, the emergence of in vitro human three-dimensional organ culture has received widespread attention because we can have more complex models which have a lot of potential uses in medicine, and I'm going to talk about it a little bit later on. So three-dimensional organoids derive either from embryonic stem cells or induced pluripotent stem cells. They can self-renew, they can self-organize, and they can exhibit multicellularity and function similar to those organs they derive from. Both pluripotent stem cells and adult stem cells derived organoids can form 3D organ-like structures and to some extent function as those organs. However, we can't just grow a liver or a stomach in a culture. There are great differences between organs and organoids. So let's start with pluripotent stem cells first. The first publications were on brain organoid, like spheroids, were reported in the 2008s, roughly. And the main problem with pluripotent stem cells is that the creation of organoids takes several months. It's a long and tedious process, and you need a specific cocktail of growth factors that have to be added at various steps in order to create those 3D organoids. The other way, so the first thing that happened was that we used patient-derived somatic cells, which were reprogrammed into induced PSCs and then expanded and then differentiated into specific cell types. Those PSC-derived organoids lose their ability to further expand, so they don't grow indefinitely once they reach the point of their terminal differentiation. And as I've said, the pluripotency leads to fairly complex organs, so they just don't produce epithelium, they produce various cell types, mesenchymal cells, epithelial cells, and even endothelial cells. So PSC-derived organoids are good models for studying organogenesis, so how organs develop during the creation of a human body, because their forming process only occurs during embryonic development. This is just an example to show you. So when you take either an induced stem cell or an embryonic cell, you take the pluripotent stem cells, add growth factors. Well, it's more complicated than that. And then you have a differentiation, and depending on the cells and on the activation factors, you get organoids for various organs. And this is another schematic. It shows you that you have endodermic cells, mesodermic cells, and ectodermic cells, and they can create various structures. Adult stem cells are different. They appeared a little bit later than the PSC-derived organoids, and they are directly obtained from regenerative human adult tissue and with a much simpler protocol than pluripotent stem cells. They have a restricted potency, so you can't grow, let's say, brain tissue from an intestinal adult stem cell, but they can lead to a single epithelial cell type depending to the tissue it has been taken from. However, those adult stem cells derived organoids provide a much better overview of tissue repair and also a better model for infectious, mostly viral infectious diseases. So they have different applications than pluripotent stem cells. And the resulting structures from adult stem cells can be expanded for a long time while maintaining genetic stability. So they don't change over time. And so they are a good basis for the expansion of cells from individual patients or normal groups, and can be used, for example, in the research of new therapeutic strategies. So again, a cartoon. So we have a tissue-derived stem cell, let's say from the stomach. They are propagated in vivo, and the result is an organoid resembling the tissue the stem cell was taken from, let's say, the stomach. So via the creation of tissue conglomerates based from adult stem cells, we can create organoids. So when we compare pluripotent stem cells to adult-derived stem cells, as I've said before, those are complementary tools. There is not a better or worse one. And they have different applications when it comes to scientific research and another new term, personalized medicine. And a variety of PSC and ADSC-derived structures have been created in a dish. Coming to the GI tract, I'd like to start with the liver. As you all perfectly know, the liver has many essential biological functions. As a gastroenterologist and hepatologist, I always say it's more important than the heart. We have metabolism in the liver, we have detoxification function of the liver, and as you know, we see that in our patients, it has a remarkable ability to regenerate upon serious damage. Major functional cell types in the liver are the hepatocytes and the cholangiocytes, and the first trials go back to the early 2000s, and most models that have been published today are either to study liver regeneration after injury. For example, there's mouse studies with hepatocytes and organoids where proliferative damage responses for a long period of time were studied, but also other applications. GI organoids, the primary function of the GI tract, as I've said, is digestion, absorption, excretion, and protection. And the most important parts are obviously the stomach and the intestine. The first intestinal organoid model, as I've said before, results back to 2009. It was based on a mouse data, and those cells, those adult stem cells from the mouse, were cultured with various growth factors like EGF or BMP inhibitor Nodgin. And 1 year later, the first gastric organoid culture system was published. And again, this derived from gastric cells. And again, you have to add a lot of growth factors, in this case, for example, a gastrin, to induce growth of organoids from those individual adult stem cells. Organoids produced from adult stem cells consist only of epithelial cells, unlike the pluripotent stem cells we take, for example, from embryos. And to obtain culture models with more cell types and various layers, we have to use pluripotent stem cells, as I've said before. Gastric organoids can also be generated directly from samples of human gastric body, for example, and those, of course, are adult stem cell models. Besides epithelial cells, and as I said before, the main difference from pluripotent stem cells is that they also contain mesenchymal cells, and so they don't only produce epithelium, and they have more cell types, and but the absence of adjacent cells, the absence of neural cells, the absence of vascularity is currently the main limitation of those models. However, new technologies are developed. One of the most promising is 3D bioprinting. You probably heard about 3D printing, where you can print whatever you want based on 3D models. And currently, something similar is tried with stem cell growth factors to create three-dimensional structures, three-dimensional organoids. This is just a short overview when we're using tissue-derived epithelial cells, the advantages and disadvantages. So as I've said, adult stem cells, you have don't have mesenchyma, you only have epithelium. Those are the limitation. And pluripotent stem cells, the main limitation that they are very difficult to culture, and they have a fetal-like character. So if you have a pluripotent stem cell, that's not a good model to study adult diseases like Alzheimer's disease. If you have a pluripotent stem cell, you can study the neurological development, but it's not a good model because of the early stage of the development for, let's say, degenerative diseases like Alzheimer's. So coming back to the GI tract, applications. In the human body, very important interactions exist between microbiota and the human body. The microbiota resides in the mucosal surface of the gastric or the intestine and have a great impact on the GI biology of the host. They can affect immune hemostasis. They participate in competitive inhibitors, so the good bacteria make sure we don't get infected by the bad bacteria. And as we also know, they greatly affect drug pharmacokinetics, and we've heard about response to therapies earlier and about drug therapeutic effects. Intestinal bacteria like lactobacillus could also promote organoid proliferation. Currently, there are ongoing studies to see if by adding bacteria from the microbiota, we can somehow protect our organoids, for example, from inflammatory factors like TNF-alpha. There are studies on co-culture, so cultures between stem cells, adult stem cells mostly with pathogenic E. coli, to study the effect of bacterial pathogens on the intestinal epithelium to better understand inflammatory diseases of the epithelium, and they're also studying studying the effect of H. pylori on gastric cells, and they're all done on organoids. And one of the earlier studies injected H. pylori into organoids and found mechanisms which lead to damage of the mucosa. And I think even more importantly, the second study on this slide showed how gastric cancer is induced by CAG-A and beta-catenin signaling in gastric organoids. So we can take those organoids as model and study the development of disease, in this case gastric disease or gastric cancer. In vitro organoids recapitulate the principles of organ biology, but offer a simplified and easier accessible cellular model that mimics specific aspects of the 3D architecture. So organoid technology may feel a need in medical research and has a potential wide range of translational applications. And here are some of the most important ones. Organoids are used today for drug discovery. You can test a much higher amount of substances for potential effects on organoids than you can test in animal models or in human studies. You can also safely test toxicology on organoids, which would be non-ethical and non-possible in humans. You can test or study the response of therapy to cancer. There are techniques of gene repairing. Transplantation therapy is a topic, and I'm going to talk about that. And obviously, you can also model genetic diseases. To start with the latter, there are currently two primary applications of organoids, and they're the modeling of human genetic diseases and the development of new therapeutic strategies. In this study, I'm citing intestinal crypt organoids were established using adult intestinal stem cells, and they were used to study cystic fibrosis. And so by having a model where you can study in vitro the disease of cystic fibrosis, you can accelerate the diagnosis, you can perform functional research, you can do drug screening, and you can start looking for ways of personalized medicine. Another issue is Oh yes, I'm slow. Another issue is individual diversities. We've talked about in response to anti-TNF alpha or the other immunosuppressants. Well, not all the patients respond as good as others to the same therapy. So we have unpredictable outcomes and time-consuming drug testing. If we have 3D organoids from one patient, we might have a tool to test in vitro prior to therapy if the patient is likely to respond to a certain therapy. And this has been done, and this is done, for example, also in colonic disease, like here, FAP. And certain compounds are researched which can be used to treat those patients to stop overproliferations in vitro. Another advantage is high throughput drug screening. I already said that. So in a much shorter period of time, you can test for more substances for their therapeutic effect. Some of them already led to clinical outcomes, like this study on antidiarrheal drugs. And also importantly, as I've said before, you can assess the toxicity in a preclinical trial of a drug, because you're testing on organoids which resemble human organs, and this is a much more reliable tool than using animal models, for example. So this is a promising tool also to test for long-term toxicity, because you can keep organoids alive for a longer period of time and expose them to a certain drug and see if there's any long-term side effects. Cancer is probably one of the most interesting applications of organoids, because the organoids have deepened the knowledge of the development of cancer, of signaling pathways in cancer, as we can create organoids which mimic tumor characteristics by taking stem cells from cancer models. So recently, adult stem cells technologies succeeded in transforming cancer tissues which have been cultured in vitro as cancer organoids. Clinical applications are, for example, response to therapy, and I'm going to show you one in the next slide. Here in this slide, H. pylori was used in co-culture with cancer cells, and to see the effect of the presence of H. pylori on the development of gastric cancer. Organoids in cancer lead to personalized medicine. So for many cancers, traditional cell culture methodologies are unable to sufficiently model the biology of native tumors. And so we have a high failure rate of preclinical compounds in clinical trials. Think of pancreatic cancer. Many compounds have been examined. Still, there is no breakthrough compound. And given the superiority of in vitro derived patient-derived organoids, and the rapid growth and the stable differentiation and the capacity to capture patient and tumor type diversity, organoids could potentially be used to identify effective therapeutic strategies and leading to a personalized therapy. What does that mean? Cells are taken from your cancer, stem cells, and organoids are created, and then various compounds are treated on the organoid, and you only receive the therapy which has been proven to work. And this has been done, for example, for chemoradiation. And so right now, many companies, many pharmaceutical companies are creating large biobanks of organoids from various tumors to look for new substances used for cancer treatment. And due to this organoid drug sensitivity testing, the most suitable drug for patients can be quickly detected. There is no clinical trial needed in the patient, so you don't have to rely on first, second, fourth line, whatever, third, fourth line therapy. And this strategy might enable the ideal treatment schemes to be selected. And there are several studies, as I've already mentioned, most of them for rectal cancer, where organoids have been used to predict response in radiotherapy. And as I've said before, that's why biobanks are created where cancer cells are cultured, organoids are created and then exposed to various chemotherapy or radiochemotherapy regimens. Another thing is drug candidates. Think of the case of ulcerative colitis Dr. Ali showed. So wouldn't it be great to take biopsies during colonoscopy, harvest adult stem cells from the colon, and then based on the organoids created from then expose it to various, let's say, biologicals and choose the right one instead of trying infliximab, vedolizumab, ustekinumab, and tofacitinib. So this is, I think, also for non-malignant diseases, a very important approach or potential approach for personalized medicine. We are not that far, unfortunately, so we're still far away from these days, but the first data is appearing, and as there's the most money in cancer, that's where the first studies come from. Gene repair might also be an interesting application, but I think we are still further away from that. Gene repair treats various diseases like cancer, diabetes, heart disease, by altering the target genes inside the body cells. So here we have really some quite dramatic interventions in the human body. And that can be based on organoids transplanted into damaged tissue and perform gene repair. So the first theoretical studies have been quite positive. As I've mentioned before, cystic fibrosis has been studied most intensively for this application, and this is a study that the CFTR function was corrected after the mutation was corrected by the introduction of organoids. So this might again be for the future a very interesting approach. Next very interesting topic, transplantation. Transplantation is essential for treating patients with final end-stage disease, let's say liver. However, there are many challenges. We have graft rejection, we have a shortage of organ donors, so a lot of unresolved questions. The organoids are infinitively available, so we can create as many organoids as we need, because there's no limitation, it doesn't depend on organ donors. And so organ donors have become potential donors. The problem is that obviously organoids are not organs. It's not that we can grow a complete liver in a lab, but it is the first step. So for instance, there's a study where they transplanted intestine organoids in a host mouse, so we're not in the human yet, and the transplanted organoid showed marked expansion and maturation of the tissue. There are studies from retina, again from the mouse, where they transplanted sheets of retinal organoids, and they could form synaptic connections, so certain functions of the retina could be regained by transplanting organoids. So although transplanted organoids are relatively immature compared to the host's organs, they can be or might be a meaningful therapeutic alternative to organ transplantation in the future. I'm approaching my conclusions. Human organoids hold great promise for applications ranging from basic research to biomedicine. Disease modeling using organoids has naturally been associated with preclinical testing in the drug development stage and high throughput drug screening using organoids, and they have been highly utilized in the field of cancer research. In addition, cancer organoids may be potential tools for identifying effective drugs in cancer patients, but not only in cancer patients. And we can also use those systems for drug toxicity and safety testing because they're so effective. Moreover, 3D organoid technology have provided attractive platform for studying host pathogen interactions in infectious diseases, most importantly in viral, protozoan, and bacterial diseases. However, so far I talked about the good parts. There are limitations. So you can't just put human cells into a piece of glass and in saline and they grow, so you need usually a mouse-derived extracellular matrix substitute as matrigel. That's one of the ones you can buy for your lab. However, first, those are biological materials, so there is variability due to the batch production. So not all the experiments can be replicated because they use a different batch. Also, if you can't clinically transplant something based on a mouse-derived matrix into human because of immune response issues. So this has to be addressed, and one of the solutions could be clinical grade collagen, and other bioengineering strategies will also improve that. Another factor limiting the use of organoid culture methods in regenerative medicine is the inability of these approaches to model multi-organ pathologies. Many diseases affect more than one organ. Again, think of IBD. You might affect the GI tract, but also the skin, arthritis, and other disease effects. So it has been tried to combine HPC-derived intestinal organoids and neurocrest cells, and the most promising studies are probably the ones on organ-on-a-chip technology, where you generate 3D systems that enable connection and communication between multiple preformed organoids. Tissue maturation is the third problem we're currently facing. It restricts organoid technology from reaching its full potential for preclinical and even clinical application. Organoid culture systems replicate the fatal brain development, for example, to a large extent if you use pluripotent stem cells. But as I've said before, it's difficult to model neurological disorders that affect the adult brain because we are using PSCs. To overcome these barriers, many protocols have been optimized to include pre-treatment of organoids, or to use certain compounds to accelerate maturation. Another big problem is oxygenation and nutrients. There is no vascularity in a organoid. So you can grow organoids up to a certain size, but then they die because they don't get enough oxygen and nutrients. And that's a severe problem nowadays. And long-term culture of organoids does not expand along with time, but rather leads to apoptosis and necrosis, as I've said before, due to the problem of nutrition. So this sums up the current perspectives, challenges, and possible solutions. And with that, I come finally to my last slide. I thank you for your attention on the evening. I thank Mazen and his team, everybody on his team at Kasaralaini for the friendship, and thank you again for having me so many times.